Welcome to the wrap-up of Chapter 18. So we left off with eukaryotic gene expression, and we were basically talking about how it's more complex than with prokaryotic gene expression. We don't have operons. Instead, we're going to have a lot more fine-tuned control over individual genes. So typically, most of our regulation is going to come in here at the transcription level, where we're just going to prevent there being the transcription factors or the other particular molecules that really, say, work. Genes by default are generally going to be off, or at least they're going to have very little transcription. And so if you just kind of leave a gene be, it tends to have very little, if any, transcription. And so our first step is just to make sure we leave it that way, or in some cases we can bind things to make it more permanent that it stays in that kind of off position. Now, if we do decide to go ahead and we produce the transcription factors, we go through and we turn on a gene, that allows us to go through and make the primary transcript. But we can affect how we cut up that primary transcript. We've talked about the splicing idea, and so based upon how we splice it, we can get different products. So if we wanted, we could adjust how we cut it up, essentially, to get a different final mRNA, if you will, that codes for a different polypeptide, and so we could still keep the gene effectively off for one particular polypeptide, even if we went through and did transcription. Now after that, we could go through this process of just not letting it out. So this would be a lot like if you have that daughter and you lock her in the room because you don't want her to date. And so it's that kind of a mentality, the Rapunzel idea, that we can just go through and make sure that the mRNA doesn't leave the nucleus, because as long as it doesn't leave the nucleus, it can't meet a ribosome, it can't fall in love, and can't make us beautiful polypeptides. Now if it does happen to get into the cytoplasm, if it does go through with this process of translation, we've got two kind of options. First, we can destroy the mRNA to make sure it stops doing so. That way we're not going to continue to get proteins. And the second option is, if we have a protein, we do still have the ability to either A, deactivate it. You'll remember that some things we can produce an inhibitor for, and that can essentially shut off the protein. Or we can go through and just cut it up and destroy the protein. So either of those would still prevent us from ultimately getting to the finish line. They would just be one of the most inefficient ways that we could possibly do it. If you don't want to go to the movies, don't buy a ticket, don't drive there, and then while you're walking through the doors, decide you don't want to and run out and leave. So we're typically just going to go and cut this off at the whole don't buy tickets, just avoid spending any money rather than going through part of the process and just changing it later. But if you did turn a gene on at some point, and you now have to turn it off, these other ones will be useful because the environment does change. And so you will have to cope with change. And so there's nothing wrong with having these methods to break down the process further along, because that does happen sometimes when you did what you were supposed to, made a gene, made a product, but now ultimately you've realized that, that wasn't that smart of an idea, and so you just have to roll with it. Now, epigenetics is going to involve three basic ways of controlling the genome from above the genome. The first one that we have will be called histone acetylation. So I will write this incredibly badly. And an acetyl group is just essentially two carbons and some hydrogens, and we can attach these to histones, and it tends to kind of push everything further apart. So it kind of makes sure the histones are a little spread out, it makes sure the DNA is held out. So this kind of makes the whole chromatin, that DNA protein complex, it makes it a little bit looser. And ultimately, the more loose things are, this isn't meant to be some type of weird euphemism, but the more loose things are, the more transcription you'll get. So this is going to favor transcription. You'll see most of the things we talk about kind of favor transcription. The other idea of what we can do is we can do what's called phosphorylating, and you guys should remember this. I might be spelling this wrong, but I'm going quickly. Wow, I can write terribly. So you can phosphorylate amino acids on proteins, and this will also have the effect of kind of making everything more loose. So once again, it's going to do the same thing where it's going to increase the amount of transcription because you're making the DNA more readily accessible for enzymes and transcription factors. And then the last one is going to be the oddball. So this will be ultimately methylating, adding a methyl group, which is just a CH3, to DNA. And so this one is going to be more of an off thing. If I put on methyl groups, I'm normally going to block the DNA, block the access of stuff to DNA, and so that is going to effectively shut the things off. But not only that, when you methylate something, it's going to be more of a long-term thing. This is what leads to stuff that we've talked about before, such as imprinting. So if you guys remember where it mattered who you got the chromosome from, whether it was mom or dad, because some of these methylation patterns on the chromosome can be passed on from one generation to the next. So we watched the video about like the fat rats that weren't licked enough when they were, oh no, those were the angry rats, that weren't licked enough when they were children, or the rats that had a poor diet that turned off a gene and got all uber fat. We watched stuff about where if you don't feed your preteen sons, ultimately your grandsons can live longer. So there's a variety of things we watched and we talked about, and there was this long-term impact of gene expression, and this is really what you see with methylation. So it turns things off, and this is one of the more permanent things. Now it can change, obviously, because it's able to kind of record info about your surroundings, but a lot of times it has certain periods in your life where it seems to be susceptible to being locked in as methylated or non-methylated, and then it'll tend to stick with that unless something changes either somehow in the far future or in the next generation. So don't be surprised if you see that. Now, this is just a picture, and it kind of gives you this idea that when you have these acetyl groups, these kind of markers, or phosphorylation, it kind of spreads things out. And you can see how the DNA is so exposed. It's just hanging right there for whoever wants you to come along. Whereas we can also have where it binds up more closely, and so you can see if it binds up, if it gets closer together, it's very difficult to get at it. It's very difficult to kind of find your way in there if you're an enzyme and grab hold of the DNA. And then we've got these methyl groups, so you can think of them as kind of like putting these bumps or markers on the DNA. So if we've got these methyl groups, it makes it hard for anything to reach it because we've kind of got like this protective covering, if you will. So it has the effect of kind of dampening things because it makes it more dense around where that DNA is. It makes it more cluttered, so it makes it harder for things like transcription factors and RNA polymerase to attach and allow transcription to occur. Now, for Eukaryotic Gene Organization, we will have these guys called control elements. Now, these guys are not transcription factors, really. What they're going to do is control the binding of transcription factors. And so these will either be what are called proximal ones, which we'll talk about coming up, but that's close to the promoter, or they will be distal, which will be further away. And so these distal control elements will contain these enhancer regions, which are spots, that will have particular activator proteins that can attach. And so when these activator proteins attach to the enhancer, there'll also be a DNA bending protein that will attach here to bend this over. It's going to have the effect of bending the DNA so that this region, this distal region, is now next to the gene. It's right next to the promoter. And so you can see that in the bottom where it's reaching the same location. And then if these activator proteins are attached, the transcription factors bind to these activator proteins. So it allows for those transcription factors to build up this complex that allows for RNA polymerase to attach. So we're going to have the triggering of the activators that bind to these enhancers. We're going to have specifically two proteins made that bend this distal region so it's now close. And because these activators are there, it's going to allow transcription factors to bind, which allows RNA polymerase to bind, which allows for transcription. So there's a lot of pieces that go into controlling this, but you can see how if we get some of these pieces, if we can kind of turn on these activator proteins to attach here to these regulatory guys, once we start that process, the rest of it can kind of cascade. It allows the rest of it to occur step by step by step and allows for us to get transcription. So this will be another way of turning things on. And so we already said that we do need these transcription factors. We have to have them if we're going to get transcription. So this isn't like optional. The only difference is, are we going to use proximal ones, which are the close ones, so these are the ones next to it, or are we going to use these distal ones, these ones that are far away, that have the added thing of having to bend to actually get brought near the gene. So these could be located thousands of nucleotides away from that gene, versus these guys are probably going to be pretty close, probably either right next to it or within like tens of nucleotides, to make sure that they're in the near area. And so if we get the right pieces, if we have the right enhancer regions that bind the right activators, that allows for us to bind the right transcription factors, and that will ultimately turn the gene on. All right. Now, RNA processing, just because we've made RNA, we've talked about the fact that we can kind of play around with it a bit, and we call that alternative splicing. So when this spliceosome goes through, you can think of the original primary transcript, so the original mRNA as is, as having a bunch of different regions. And so as this spliceosome goes through, it's going to cut out some of those regions. But we don't know which ones. It's kind of like getting a pizza menu where it's got just a list of like 30 topics. 30 toppings would be correct there. And so as you go through, you can kind of say yes, no, and determine what actually gets produced. You know, what's actually part of that finished pizza. And it's the same idea here where the spliceosomes can go through and cut out certain sections, so it can cut out these darker ones, and so they no longer become part of our finished transcript. Now, if you cut out different things, you can get different results from the same original possibilities. So when you talk about the pizza analogy, if I go through on one and get rid of all the vegetables, I get a delicious, meaty pizza. I could go through with the next one and cut out all the meat type pieces, if you will, all those options, and just get the vegetarian ones, and you would get something that's not really pizza, because it's vegetarian, and I don't believe that's a real pizza. And so at this point, we can see that one gene can have multiple different alternate possibilities based upon this processing step. So just because I go through and actually activate a gene or transcribe a gene doesn't necessarily mean I'm going to get what you're looking for out of it. It could be I activate the gene and produce a completely different polypeptide that's one of the possibilities, and so the other possibility is still effectively shut off. It just depends on what we do with it. And then lastly, we have pattern formation. So this is how our body determines spatially where everything goes, to make sure we put the arms where the arms should be and the eyes where the eyes should be, the knees where the knees should be, all the pieces. You know, you can recite the songs, head, shoulders, knees, and toes, whatever. It does this by using something called positional information. And that's where our body has, where it's able to detect kind of what's up, what's down, what's left, what's right, what's forward, and what's back. These are the simple terms. You typically say ventral and dorsal and all those fun terms. But our body can figure this out. So in this XYZ arrangement, we know where things are at. And so this allows for us to affect gene expression. So we will have certain things turned on, certain chemicals will be produced in certain regions, and this is showing the different chemicals in different places in different development of fruit flies. And so in that embryo, we're going to kind of affect where these chemicals are at. And as these chemicals, which we can say are cytoplasmic determinants, if we will, that's a simple way of doing it. So these chemicals then, based upon where they're at, will help determine gene expression. And so we're going to look at our body's capable of kind of detecting where something's at, and in many cases it can detect the movement of cells or trigger the movement of cells. So you'll see a lot of times cells will migrate, and it's a migration that actually forms stuff like our digestive tract, where we have this kind of ball of cells, and then some of them start to migrate and form a pore, and then that continues till you get a tube, and so we now have kind of a ball with an entire hole through it, and that hole ultimately becomes your digestive tract. And so it's not just about kind of which genes do we activate and just about where a cell's at. Part of it too is where do I move things and where is something moving to that helps determine which genes overall get turned on fairly permanently. We call that cell differentiation. So this isn't like a really, really temporary thing. This is a much more permanent thing. And based upon the differential gene expression, that determines what type of cell you are. And so if I want to go through and turn on a specific subset, kind of like we talked about in the library, where if I focus on things like detox and controlling sugar, whether it's storing glycogen or releasing it, I could end up being a liver cell, which I shouldn't be touching my arm for, my liver cell. And so that's what you'd see just in the liver. And so that liver cells, in animals at least, typically permanently activate certain genes that makes it a liver cell and permanently deactivates ones that would make it like an eye cell. Now there can be some fluctuation over time with other genes, but it's a fairly permanent thing when you differentiate. Now plants you'll see are a little bit more wishy-washy on the differentiation thing. That's why you can take clippings of plants, put them in water, and you can see sometimes where like the bottom of the clipping will develop into roots because the cells can change. They're not fixed. You know, and there's a lot of research being done in humans and animals to see if there's ways that we can do this with our cells, where we can kind of trick a skin cell into thinking I'm a kidney, because that way I could potentially take some skin from you and grow you a kidney. So things like kidney cancer wouldn't be such a big deal anymore, because you could just be like, I'll make more. So this process all comes down to, though, what genes get activated during this differentiation process, which typically occurs very early in the animal's life cycle. So when you're an embryo, you're already starting this differentiation process, and it continues until you get to like a fetus, where within the fetus it's kind of differentiated. It just has to grow. It just has to, elaborate to get bigger, to get more capable. But you still have the beginnings of lung tissue, even in a fairly early developed human, or bear, or rat, or whatever. And then from there, they just continue development until they're functional. And then at that point, you're born, everything should go well. Try to make sure you don't lose any important organs, because you cannot grow them back as we have it right now. And we cannot convert something else to become one as we have things right now. If you develop that, you will be rich and you'll get a Nobel Prize. The last slide, just because it's awesome, Antennapedia. So Antennapedia is where we trick things and we say, okay, what we're going to do is we're going to try to rearrange some stuff. And so some of these genes we have tell us where things go. If we mess with those a little bit, we can confuse things. So we say, where would be a good place for some legs? Where your antenna go. And we get Antennapedia. Pedia for foot or leg. Antenna because it's where its antenna is supposed to be. I don't believe these guys are any more adept at walking or crawling. The fruit flies with legs on its head. I don't know. Maybe the ladies will like it. I don't think so. This is not ultimately saying that we can easily go through and do some Mortal Kombat stuff where we've got like 6 or 4 arms or whatever. This typically doesn't end well, so we're not probably going to experiment too much on people. But we have experimented on stuff like fruit flies to show that this does pan out. If we can disrupt things early on in development, you can confuse the body, and so the body still knows how to make a leg. It just has to figure out where to put them. And sometimes they'll get put in the wrong spot. So congratulations, Mr. Fruit Fly. You have a beautiful set of legs. Take it easy.